Hello, good morning, everyone. Myself, Dr. Ravi Kumar Gupta, Assistant Professor in the Department of Environmental Microbiology at Babasaheb Bhimrao Ambedkar University. Today, I am going to discuss about the most advanced molecular tool for gene editing, that is CRISPR-Cas system. Gene editing is the insertion, deletion, or replacement of DNA sequences at a specific locus in the genome of an organism. Artificially engineered nucleases are generally used as a tool to achieve gene editing by introduction of double-strand breaks at the target site. However, recently developed CRISPR-Cas system has made commendable success in gene editing of human genetic disorders. CRISPR-Cas system has potential role in diverse scientific fields such as agriculture, environment, and human health affected by familial or non-familial diseases. Interestingly, absence of CRISPR-Cas system in eukaryotes make it powerful tool for gene manipulation and editing in human. Due to its low cost, easy manipulation, and a straightforward mechanism, researchers find CRISPR-Cas system as the most advanced tool for gene therapeutics. Several clinical trials for the cure of human genetic disorders using CRISPR-Cas system are completed or underway. Although there are some challenges with this technology, such as delivery of foreign DNA in human body, host immune response, and risk of off-target mutations. However, I am very hopeful that in near future, scientists all over the world will find solutions for these hindrances, and we can easily cure the human genetic disorders. In this lecture, we will study what is CRISPR system, mode of action of CRISPR system, genome editing with CRISPR-Cas system, and application of CRISPR-Cas in genetic disorders. So what is CRISPR system? Clustered, regularly interspaced, short palindromic repeats, termed as CRISPRs, were discovered in prokaryotic genome as a repetitive sequence and functionally responsible for the conversion of alkaline phosphatase. Later on, discovery of CRISPR-associated sequences termed as Cas protein, which work as nucleases and help to cleave complementary sequences of bacteriophages, form the CRISPR-Cas system. Till now, CRISPR-Cas system is the most advanced tool for gene editing and used for therapeutic interventions to treat human genetic disorders. Increasing human population and anthropogenic activities in the destruction of environment create selective pressure on the environment and human health that may alter the human gene pool. This may eventually lead to the generation of various human genetic disorders. Hence, conventional and traditional approaches for the treatment of these human diseases will be no longer sufficient, and we need to develop new approaches for the betterment of human health. Theoretically, gene editing is not a new concept because our cells do this job almost thousands of times in a day to repair our DNA through DNA repair mechanism during replication and makes our genome integrated. Many endonucleases are programmed to alter the DNA sequences and led to the discovery of meganucleases, zinc finger nucleases, transcription activator like effector nucleases, and CRISPR associated nucleases. CRISPR-Cas system is gaining popularity day by day because Cas enzymes are RNA guided. They work on Watson-Crick base pairing and can be programmed to alter the DNA sequences according to our need. Whereas other endonucleases work on protein DNA interactions. CRISPR-based therapeutics approaches are widely accepted because of its broad applications as a gene editing tool and clinical corrections of human genetic disorders. Although CRISPR-Cas system has great potential as a gene therapeutic, however, it might not work in some genetic diseases. For example, inserting and replacing a mutated gene copy with the corrected exogenous gene copy might not produce the regulatory elements required for the proper expression and function of a gene. More research needs to be done for the improvement of current technologies. Since CRISPR-Cas system is not present in eukaryotes, scientists use this genetic tool to edit, delete, add, and modify diseased gene in humans. Enzymes are RNA guided. They work on Watson-Crick base pairing and can be programmed to alter the DNA sequences according to our need. Whereas other endonucleases work on protein DNA interactions. CRISPR-based therapeutics approaches are widely accepted because of its broad applications as a gene editing tool and clinical corrections of human genetic disorders. Although CRISPR-Cas system has great potential as a gene therapeutic, however, it might not work in some genetic diseases. For example, inserting and replacing a mutated gene copy with the corrected exogenous gene copy might not produce the regulatory elements required for the proper expression and function of a gene. More research needs to be done for the improvement of current technologies. Since CRISPR-Cas system is not present in eukaryotes, scientists use this genetic tool to edit, delete, add, and modify diseased gene in humans. Now, origins of CRISPR-Cas system. The discovery of CRISPR happened accidentally in 1987 by Japanese scientist Yoshizumi Ishino during the study of phosphatase enzyme in E. coli. These spacer sequences were further used for strain identification in Mycobacterium tuberculosis by von Ambedkar and his co-workers and named it polygotyping. Eventually, many repetitive sequences were identified in various archaea and bacteria and thought to play an important role in DNA repair mechanism. It was found that bacterial strains, which consist these homologous spacer sequences similar to the sequences present in bacteriophages, are immune to the infection by bacteriophages. This study suggested the role of CRISPR-Cas system as a defense mechanism and gave adaptive immunity to bacteria. Advancement of genetic tool lead the researchers use the CRISPR-Cas system for gene editing. There were several genes identified which encode DNA repair proteins associated with CRISPR and termed as Cas genes, which encodes Cas proteins. There are 4 Cas genes discovered in prokaryotes, named as Cas1 to Cas4. Cas1 and Cas2 did not present any similarity to known proteins, but Cas3 resembles to helicases and Cas4 belongs to REC-B exonucleases. Till today, total 7,014 genomes are analyzed and 8,939 CRISPRs are found, in which 870 CRISPRs are present in archaea and 8,069 are present in bacterial genomes. Now, classification of CRISPR-Cas system. In early genomics era, many CRISPR locus were identified in archaeal genome, specifically in thermophilic and hyperthermophilic organisms that suggested its role in adaptation of these organisms at high temperature. But later on, many CRISPR locus were identified in mesophilic bacteria that revealed some different function of this locus. Scientists identified that spacer sequences in CRISPR locus were adapted from bacteriophages and prophages. This was the first evidence for CRISPR involvement in bacterial immune defense mechanism. The universal immune defense mechanism was present in most of the bacteria and archaea. Comparative genomics reveals substantial differences in Cas genes and effector molecules that create diversity of CRISPR system. In any given genome, the number of CRISPR array may vary from 1 to 18, and repeat sequences present in CRISPR may be from 2 to 374 in numbers. It is to be noted that some closely related bacterial strains have substantial differences in their CRISPR content, and few distinct bacterial strains may have almost identical CRISPR content. For example, Mycobacterium tuberculosis has distinct CRISPR sequences, but Mycobacterium leprae did not report it to have any CRISPR sequences. Furthermore, E. coli and Mycobacterium avium have almost identical CRISPR content. The classification of CRISPR-Cas system is based on the encoded effector protein, that is, Cas proteins, in different stoichiometry. Typically, CRISPR systems have been divided into two major families, namely Class 1 and Class 2 CRISPR system. Class 1 system have multi-effector complexes with 4 to 7 Cas proteins, whereas Class 2 system have single multi-domain effector protein. As you can see in the figure 1, the majority of archaeal organisms consist Class 1 CRISPR-Cas system. However, most bacterial strains contain Class 2 CRISPR-Cas system in nature. Based on the organization of the Cas genes and signature sequences present in it, the two classes of CRISPR-Cas system consist three types, namely type 1, type 3, and type 4 in Class 1, and type 2, type 5, type 6 in Class 2 CRISPR system. Thank you for your patience.